You can go ahead and get started whenever you like.
Okay, great, thanks. I'm just gonna close you guys off a little bit. So thanks for coming or watching this video later on. So I don't know how much you have heard already about some of the things that I'm going to talk about, but I guess if you've heard them twice, that means, really pay attention. They're important. So first I'm going to talk a little bit about what we do in my lab, and then share some things that something I'm involved in that's bigger, that's regional, and then just give you some advice if you want to get into research during your undergraduate degree. So my research is, if I put it in the big picture, is asking about how do cells know what they should do. So I look at cell-cell communication. So cells communicating to other cells and asking about how they're receiving the signals, how they're interpreting those signals, how they send out those signals, and then the consequences of those signals. So in diseases like cancer, some of these signals are messed up and cells either don't respond properly or they send out too many things and it makes the other cells around them grow better or what have you. So a major part of my lab works on B lymphocytes. So B cells are the cells that make antibodies, and antibodies, you probably know, are really important for protecting you against infection. But they also can be detrimental if you generate antibodies to your own self. You can get autoimmune disease. And so B cells are generated from hematopoietic stem cells. And so a stem cell is a cell that can be anything. So just like you as a first-year student, you can still be anything. So in the stem cell. But a B cell, when it starts developing, it's actually put on a trajectory to become a B cell. So we're looking at that developmental process where the B cell is making decisions about moving on in its development. Specifically, we're working on something called extracellular vesicles. So we call these EVs, which is kind of a shame because right now, if you hear EV, you probably think electric vehicle. But we call extracellular vesicles EVs. And there's these little lipid-bound nanoparticles that either are secreted from inside the cell, like here we call it exosome, or are butted off the plasma membrane, so on the surface of the cell that we call a microvesicle. And what we found is that when we stimulate this receptor that I'm interested in called CD24, it's not a very exciting name, but that's its name. So we stimulate this, we activate that receptor, and we cause release of these microvesicles. And then those microvesicles can actually transport these receptors that are on the microvesicle to recipient cells. It can change that recipient to its environment. So these microvesicles are important for this intercellular communication. The consequence of or the reason why they're doing that is still unclear, and so that's what we're trying to figure out. So tools that we use, we go into the lab and we culture cells in a dish. We manipulate those cells, and then we look at them using tools like flow cytometry or fluorescence activated cell sorting. And this is a complicated slide. Don't worry about the details or anything. But basically what we do is look at a single cell level and analyze each cell as it passes by a laser and gets the laser light gets captured, and we can know what's on that cell and how big that cell is, so we can actually track the movement of these microvesicles at a single cell level. We also use omics, so looking at all of the DNA or all of the RNA that's in a cell or in the extracellular vesicles. So for example, we could have two conditions. We isolate all of the RNA and sequence it, and then we can do some downstream analysis to figure out what's going on inside that cell based on what RNA is present. So some pretty fancy analytical techniques go into this. And it's pretty exciting. We also use animals in our research. So we have a wild type mice that has all of the genes that are normal mouse has. And then we have a mouse that the only difference is that receptor that I'm really interested in called CD24 is missing. So it's knocked out. And we can compare these two mice to each other to see what happens if you're missing CD24, because then that tells us what CD24 does normally. So that's one project. The other big project in the lab is actually about breast cancer and fat cells. So what fat cells are, they're cells that take extra energy and store it as fat. They store it as lipids. And many people have trouble with this. They store too much fat, but it's actually really important. If you don't have enough fat, you can't regulate your temperature or body functions. But we're looking at the fat as a supporter for cancer cells. Okay, so this is a tumor cell. It's just following orders. There's something changes in the genetics to tell it to proliferate uncontrollably. But the tumor microenvironment, the cells around it, actually can help that cancer cell grow and spread. And so that's what we're interested in. Again, this intracellular communication between these cells that are saying, okay, you can hang out, and the cancer cell takes advantage of that, takes advantage of whatever the fat cell can provide to change and to grow. So specifically what we're looking at is this process called metastasis, where cancer cells will leave the circulation, and then they hit a secondary site, and they change and they go from this elongated migratory or cells that move into cells that stick. And in that way, they can colonize new sites. So this process we're interested in because we have information, we have data showing that fat cells can actually promote this colonization. So we're looking at the interaction between fat cells and these breast cancer cells. And again, they're changing from this migratory form, this elongated form where it can move, it's called mesenchymal, to a form that sticks and is a little bit more round, called epithelial. And we found that there's some protein that causes this change, and this protein is secreted by fat cells or adipocytes. And we just called this protein adipocyte extracellular matrix induced protein. I have no idea what it is, but the next round of experiments, hopefully we actually identify what that protein is. So tools we use for this project is, again, cell culture. So we put cells in dishes and we grow them in the lab. And in this case, we combine those fat cells, those adipocytes, with proteins that are normally found in the environment, and then layer on breast cancer cells. So all this mix of cells has this really complicated interactions using to look at specific proteins and to look at the shape of those cells. We also will be going into mice at some point to actually follow the movement of these cells to see if they really do become more metastatic, more migratory after they've been exposed to fat cells, or less migratory after they've been exposed to fat cells. So if they colonize better. So that's my lab, big picture. The other thing I'm involved in is this Terry Fox Research Institute Marathon of Hope Cancer Centers Network. And this is a regional initiative that's bringing together cancer researchers and clinicians from across the Atlantic provinces to work together to build this foundation for precision medicine. And what I have is a short 2-minute video giving the overview of this, this vision. So hopefully you can hear it.
We now
Speak up
have
if
a
you can't
roadmap
hear
to cure cancer. It starts with our DNA. Every cell in our body contains it. It's our genetic code, the instruction manual for our cells. It tells them how to function. And like a fingerprint, it's unique to each of us. Think of it as our human operating system. However, when our cells malfunction and mutate, they can become cancer. With the mapping of the human genome in 2003, everything changed. The genetic code for each individual cell can now be identified, pinpointing exactly which cells are cancerous and targeting them. This is called precision medicine. Precision medicine requires vast amounts of data, which is why the Terry Fox Research Institute is creating a national network of cancer centers that will unite cancer researchers and top-tier hospitals, forming the Team Canada of cancer research, sharing data, detecting patterns, and comparing diagnostics, treatments, and outcomes. Each time a cancer is treated anywhere in the system, that knowledge will be added to the network. It will learn and grow. Information on how to treat all types of cancer will be at the fingertips of our clinicians, allowing them to deliver personalized treatments and therapies. The best treatment at the right time for the right person in the most efficient and cost-effective way. Terry Fox's dream was to cure cancer. He inspired us with his dream. Today, the human race is on the verge of gaining immense new knowledge and the power to heal. This is the roadmap to cure cancer.
So it's a big dream, it's a big vision, and we're just one piece of that. So there's the proposed structure for this Marathon of Hope Cancer Centers network is 5 major consortium, and we're the Atlantic Consortium. The other thing to know, especially if you're interested in cancer research, is that there's this regional institute, virtual institute called the Beatrice Hunter Cancer Research Institute, and they support all forms of cancer research. They provide financial support for graduate students, but also for students working in the summer in a research lab. They host conferences and workshops, and they promote getting together of the all different researchers from across the region. So Beatrice Hunter Cancer Research Institute is really heavily involved in the Marathon of Hope and helped bring together this Atlantic Cancer Consortium. So what are we looking at when we're talking about or what did we decide to research, I guess, in our consortium? So overall, it's interesting or maybe sad to know that Atlantic Canada has some of the worst rates of overall cancer in the country. So we have lots of new cancer cases. It's a leading cause of death. And one of the things that we're going to do is focus on a couple different cancers, but take samples from patients, take data from patients, take information about their cancers, and put this into a biobank that it can be shared across the region. And so this idea that something happens in one part of the country can affect something or can influence decisions in another part of the country really depends on being able to analyze a lot of data. Patients, so it's biobank. So this is a really big infrastructure project. Analysis of all this data is called bioinformatics, and it's really important to be able to analyze it correctly. And so we actually have a whole core team who are responsible for analyzing all the data that's going to come out of this project. And then, of course, if you can't use the data or you can't have new people who can use the data, you can't really build. So there's another project on training and education. The specific cancers that we're focusing on are colorectal cancer, and there's 3 main projects, and we're not going to get into that here. And then lung cancer. And this is because we have the highest rates of colorectal cancer and lung cancer in all of Canada, and the highest death rates as well. And all of this is going to be interacting with each other and supporting each other. So that's the idea. And so what we have, our vision is that you can, instead of just taking the patient who gets the tumor at some point, presents with symptoms, gets the diagnosis, treatment, and monitoring. So at these time points that are indicated by arrows, we can actually have additional clinical interventions, or maybe analysis of the immune system, or maybe we could do something called a liquid biopsy. So this is where you take a blood sample, and in that blood sample, you look for specific cues or proteins that will tell you that this person has cancer and how that's doing. Okay, so how does this all get funded? How does research actually happen? Well, it happens with competitive grants to the principal investigator, who's your assistant professor, your associate professor, or full professor. So this is just different levels of being a professor. And lots of different agencies will fund research. So tri-councils include NSERC, CIHR, or SHRC in the humanities. Charities will fund research. So the Cancer Research Society or the Canadian Cancer Research Society, Terry Fox Research Institute, and for example, NMUN will also fund some research. So who does this research? Well, we call you highly qualified personnel. So undergraduate students, like who work in the summer full-time, who do their honors project, or who do a work study, all contribute to research. Graduate students, master's students, and PhD students. And then the students, or after you're done your PhD, there's a training phase called postdoctoral, where you're called a postdoctoral fellow. And that's where a lot of the work, that's who does a lot of the work as well. And then we do have technicians and research assistants. So how is our trainees funded? Well, trainees are funded by a variety of mechanisms directly. So I can get my research funded so I can pay for the cells and the microscopes, but also as a trainee, you can get yourself funded. So through NSERC, there's undergraduate research fellowships. I want to point out that there's an Indigenous pool of fellowships. So if you identify as Indigenous, you have a different and a better chance at getting these NSERC undergraduate scholarships. So really do apply for that and check that box. At the MSc and the PhD level, CIHR or NSERC will give fellowships, and same at the postdoctoral level. And sometimes these are supplemented by other awards, and sometimes you can get fellowships by charitable organizations. And so these are just some funds that have funded, some sources that have funded my trainees. And sometimes from your principal investigators' operating funds as well. They can pay you. So if you're interested in research, what do you do? Well, here are some tips. So start early. So if you start looking for a summer position in April, it's too late. All of the summer positions have been taken. Start in October. Apply to programs for funding. It's easier for me to hire you if somebody else pays you. So for example, we have two programs, one for all students called the MUSEP program, and one for international students called the ISWEP program. This is in the careers and experiential program. And this happens in the first week of term, you apply for this. We have science undergraduate research awards, and applications for those come in January or February. These SURAs can be held in the Faculty of Science or Medicine, but they're actually two competitions. So you apply for one or the other. I keep saying NSERC, National Science Engineering Council of Canada, these undergraduate summer student research awards. They go through the applications again are January, February, and they go through the Faculty of Science. But if you decide there's an PI in the Faculty of Medicine that you want to work for, you're more than welcome to hold it there. You just have to declare that. And lots of first-year students get these. So they're mainly based on grades. So if you have really good grades, you should be applying. And you're interested in research, you should be applying. As you get more advanced, you can do an honors research project. So working in a lab for a full year as a part of your courses. If you are interested in, as a woman, getting involved in research and getting some more exposure to people who do research, you could consider joining WISE-NL. And if you're interested in figuring out who you might look for, the best place is actually to go to the website for the faculty, for the person, for the researcher. So you go to the department website, they always have a My People's tab. You go through, you click on all the different links, and you actually read what people are doing. Because what they teach might not be exactly what they're doing research on. And there is this site called YAFL, but I have to be honest, I've never used it, but it's also supposed to highlight the different research going on campus. So that's all I had prepared. So I don't know if there's any questions. There's only one person here, or two people here.
We had another student, but they messaged me personally that they were having some connection issues. So they might come back. We're not sure. But yeah, research is something that's super important to get into as an undergrad student. It provides so many opportunities along your degree, and you make so many connections along the way. So whether you're a first-year student or a student who's in their third year, or you're just joining the program, research is something that is super important to consider. So definitely, like Dr. Christian said, go through the My People page on the faculty's website and look for professors that are doing something that interests you and see if they're providing any MUSEPs in the fall. It's a great way to get involved with your department. So if there's no questions, we'll go ahead and stop the recording. And then we'll post this on the D2L for anyone to watch after. We've been getting a lot of questions from students after they watch it online. So any questions that I can't answer, I'll probably direct towards you, Dr. Christian, if that's okay.
Yeah, that would be no problem. And it's going to be hard to stop by my office because I have like a half moved two offices. But, you know, in the future, if anybody wants to stop by my office and have a chat, I'd be more than happy to.
Yeah, perfect. Anyways, thank you, Dr. Christian, for taking time just to share that little presentation about biochemistry. It's super, really appreciate it. So yeah, thank you so much. And then we're free to leave.
Thanks for having me.
Thank you so much.
Okay, bye.
Bye-bye.